Watson and Crick who? The woman who helped discover the structure of DNA. The discovery of the structure of the DNA double helix is probably one of the biggest discoveries in existence and is easily one of the most controversial in scientific history. In 1953, biologist James Watson and physicist Francis Crick discovered the structure of DNA, the DNA double helix. However, what is not well known is that without the help of a chemist by the name of Rosalind Franklin, they may never have made this discovery. What is controversial is that Watson and Crick may have obtained Franklin's help without her consent. In 1953, Watson and Crick published in Nature that they had successfully discovered the structure of DNA. In it, they described DNA as being a 3D alpha helix made of two chains that run anti-parallel to one another that exhibit Chargaff base pairing with respect to the bases. This means that A pairs with T and G with C. The bases were positioned toward the inside of the helix with phosphate facing the exterior. The helix was held together by hydrogen bonding between these bases that could easily separate it, unscrewing the corkscrew double helix into a ladder so that each strand could be copied during replication. For their efforts, Watson, Crick, and physicist and molecular biologist Maurice Wilkins were awarded the Nobel Prize in 1962 in physiology or medicine. But how novel was this information? Well, in 1871, a Swiss chemist by the name of Friedrich Miescher discovered that DNA was the genetic material. In 1950, a mathematician by the name of Erwin Chargaff had found that the bases existed in proportion, A with T and G with C, so the pairing wasn't like with like as Crick had predicted. Watson and Crick adopted their famous molecular modeling from American chemist Linus Pauling. There were two major teams of scientists working hard to discover the structure of DNA with what was known: Watson and Crick at Cambridge University in London, and Franklin and her supervisor Maurice Wilkins at King's College, also in London. The race to finish would be dependent on who had the best technique. Watson and Crick took the structural model approach that was not based on experimental data, while Franklin, who worked along, based her evidence on experimental data using X-ray crystallography. X-ray crystallography involves shining an X-ray beam through a crystallized molecule in order to scatter or diffract the X-rays. The rays bounce off the electron of the molecule and are captured on a film plate behind the image, producing a photograph. X-ray crystallography is very difficult and the specimen must be in good condition or the image will be blurry. At the time, another chemist named William Asbury had been using this technique to take pictures of DNA. However, his work was not that great because his specimen was not a pure mixture of DNA, but a mixture of both forms, and his equipment was old. Rosalind Franklin, on the other hand, had discovered that two forms had existed, and with her new equipment provided to her by Maurice Wilkins, had produced the most clear picture to date. Watson and Crick relied on the Asbury images of DNA, while Franklin had taken her own photos but kept them quiet. Watson and Crick, along with Pauling, had already published scientific papers with incorrect structures of the DNA helix, to which Franklin looked down upon. She believed in not publishing data until she had absolute concrete proof. Franklin had proof in her famous X-ray 51 photo. It was the clearest picture of DNA structure in existence, but Franklin never got around to studying it. Frustrated with Franklin's cautious approach, Wilkins went behind her back and showed Franklin's X-ray 51 film to Watson, who deciphered the image clearly as a double helix. They realized that Franklin was correct when she predicted that the phosphate backbone was oriented toward the outside of the structure, not towards the inside, as Watson and Crick had predicted. She talked about this discovery at a conference that Watson attended. He scoffed at this idea, and all he could say was that her appearance was shoddy. He is famously quoted as saying, "By choice, she did not emphasize her feminine qualities. There was never lipstick to contrast with her straight black hair, while at the age of 31, she dressed like she showed all the imagination of an English blue-stocking adolescent." From there, they solved the mystery, published in Nature the same year in 1953, titled "A Structure of DNA," In 1962, Watson, Crick, and Wilkins shared the Nobel Prize. Franklin had died in 1958 of ovarian cancer. Only 3 individuals may share the Nobel Prize, and the work cannot be awarded posthumously. However, if Franklin were alive, would she still have been awarded the prize? After all, she was not really acknowledged in Watson and Crick's paper. Instead, her boss, Maurice Wilkins, was. But this is not marked the first time in history that a female is overlooked, and it won't be the last. Should the panel of pictures of who discovered the structure of DNA look like this? Perhaps this. What about this? Or maybe just this? For now, it only looks like this.